Hello, everyone. Happy New Year, and welcome to our monthly Science of All Protein seminar. My name is Amy Huang, and I'm the Senior Associate Director of Scientific Ecosystems at the Good Food Institute. Welcome to our community. If you're here for the very first time, GFI is a 100% driven by philanthropy, nonprofit think tank. Our goal is to build a world where alternative proteins are no longer alternative, and science is at the very center of that, which is why we host these monthly seminars and why we build lots of valuable resources for the scientific community. If you are looking to take a deeper dive into the science of plant-based fermentation or cultivated meat technologies, check out our open access resources at gfi.org/science. Today, I'm super thrilled to be joined by Dr. Afroditi Chatzifragkou, the Professor of Sustainable Bioprocessing at the University of Reading, who will discuss how rapeseed meal, a common byproduct of oil production, can be repurposed as a feedstock for microbial fermentation. Using yeast, her team investigates how this side stream can be used as a substrate to produce astaxanthin, a high-value natural pigment used in food and other applications. Afroditi Chatzifragkou is a professor of sustainable bioprocessing in the Department of Food and Nutritional Sciences at the University of Reading. Her expertise lies in food bioprocessing. Afroditi's research focuses on microbial fermentation and biorefineries, alternative proteins like plants, insects, and seaweeds, as well as the optimization of green extraction technologies, such as supercritical fluid extraction, ultrasonication, enzymes, etc., for functional compounds and food applications. I had the unique pleasure of spending a week together with Dr. Chatzifragkou in October, and she just has such a wealth of knowledge to share and is an incredibly valuable member of the community. So I'm delighted that she's here to share her research with us all. Thank you so much for being here with us today. I will turn the floor over to you.
Thank you very much, Amy. It's a pleasure to be part of the GFI community again. Hello, everyone. Thank you for the very warm welcome and introduction. Without further ado, I would like to tell you a couple of things about the things that we do at the University of Reading and the Department of Food and Nutritional Sciences that I'm a member of. And then we're going to talk a bit about the work that we did a few years ago with regards to rapeseed meal valorization and astaxanthin. Right, so this is where we are. So the University of Reading is 25 minutes a drive outside of London. So it presents a nice opportunity to be part of a commuter's town, not a city. This is a very nice aerial image of our campus, our very nice green campus. Actually, you can see half of it because the other half is um, from the lake. I think our building is somewhere here, if I'm not mistaken, the Harry Neston building, which houses the Food and Nutritional Sciences Department. As a university, we are celebrating our centenary this year. So we essentially it's been in March, I think, at some point mid-March 2026, it will be 100 years and then we became a university and we granted a lawyer charter. So big things are coming up for the university. And another thing that is quite important is that we have, as you see, a very nice campus. So we've been recipients of a Green Flag Award over the years. And we have sustainability in the forefront of what we do for a number of years now. And another example of how we think about how we want to transform the way that we consume foods and the way we think about food and food choices is that we were the first UK university that participated in the Menus of Change Research Collaborative, which essentially rethinks how we make our menus a bit better. So this has been rolled out throughout our hospitality sector within the university, and it's an initiative both for all of our students and not only the food and nutrition sciences ones. To see how we can embed a bit more plant-based products, how we can produce food from local sources rather than spending miles to get sources into campus, as well as think about how we reduce waste and how we repurpose ingredients in foods. So sustainability is at the forefront. The University of Reading has leading expertise in the area of agriculture and food. And within that leading expertise, a couple of years ago, we sat together and we formed a strategy. So this strategy is called the agri-food future strategy, which theoretically and in practical should bring together all the collective expertise that we have in the university in collaboration with an extended network of stakeholders, with the idea that we are going to think and reshape and transform our food systems by 2015. So within that, this is our vision. So we envisage that by 2015, or hopefully a bit earlier, everyone across the world will consume a sufficient, healthy, sustainable diet that leaves the lightest possible footprint on the planet. So as you can see, there are elements here that we would like to bring together. So obviously nutrition is at the forefront and public health is at the forefront. How we produce foods, how we allow consumers to move on to a sustainable diet for the future, but also leaving the smallest footprint on the planet, thinking of how we could potentially produce food across the supply chain that it is more sustainable and of course affordable. And within that, we have three priority areas that are linked. And one of these is a bit more focused on what we do in terms of research activities in the Department of Food and Nutritional Sciences. But these are the three areas that we support. So the first one is around farming systems. So essentially how we innovate in terms of producing foods while we are protecting and enriching natural resources. The second priority area is a resilient food system, so a food system that meets our needs, but also in moments of crisis and in the face of shocks, as we have recently seen, we need to be much more resilient. And the third priority area, which is where the department pivotally sits within, and the research that we do sits within, is how we can ensure that we deliver food that nourishes people and, of course, the planet. And within that, we have a number of strengths within the department in terms of research purposes and research focus. So food science obviously sits at the forefront. Behavioral sciences also sits at the forefront. How consumers choose food, what are the drivers for innovation, for change, how they apply and how they understand sustainability, and how flexible they are with innovative food products and how we can actually help them towards that direction. And then other aspects is that, of course, we need to make sure that whatever we develop as innovative food for the future needs to be desirable. So consumers will need to come back and eat it. Otherwise, it will be a lost cause. But also affordable and accessible foods. We've been working a lot lately with a number of research projects around local communities which are disadvantaged, trying to see how we could potentially penetrate those areas and make sure that these people also have the liberty of choice towards nutritious and affordable food. And then obviously drive the shift towards healthier diets and food choices. So these are within that particular area. We work collaboratively within an environment of around 30 or so academics within the Department of Food and Nutrition Sciences. This is everyone at the moment. So we can see we have different areas of expertise. We are a very amicable department. We like to work together. The drive of science is very much multi and interdisciplinary these days anyway. But we offer a breadth of food systems expertise, so from post-harvesting produce up until the point of nutrition, influencing diet, improving health, but also in terms of gut, cognitive function, and so on. Lots of areas that are interlinked in one way or another. So we have a couple of focuses around here as a department. So from a food science perspective, we focus on innovation of foods that are produced within low emission activities as well, and how we produce efficiently products within a circular economy and how we reduce waste in that matter in order to obviously support sustainable and healthy diets. And then from a nutrition perspective, we're looking into how we can profile sustainable food products and then obviously see what sort of nutrients and non-nutrient components they have and how they impact health. Qualifying impact health normally through human trials and see obviously how the shift in certain diets affects health. We like to see food and diet as a way to prevent disease, obviously, and we have specific focuses on non-communicable diseases such as cardiovascular diseases and so on. And then the other thing that we do here in the department and within the UK generally is how we can influence policy, because we need to make sure that whatever evidence-based research that we have is get fed upwards toward policy making that will protect the public health and will drive all these innovations forward for people in general to be able to benefit from. Right, so a couple of things about me and my research interests before we start with the actual research topic. So I have to say that fermentation sits very closely to my heart. I fell in love within the first couple of years as an undergraduate student, and I've been in love ever since. So I've worked with all types of bacteria apart from algae, all types of microorganisms, apologies, apart from algae. I'm afraid they have escaped. So mainly wild type grass status ones and a couple of engineered strains that we have worked over the years. And the interest on microbial fermentation is quite broad, so from lipids to pigments to proteins to polyhydroxyalkanoates, bacterial cellulose, and some bioplaced pathogen chemicals, so food and non-food applications across the years. And we have also looked into how we could potentially use wastes and residues as fermentation substrates to drive a bit down the costs for fermentation. And then another part is focusing on new proteins. So we've been working with seaweeds, edible insects, primarily crickets, but also plant-based proteins, trying to see how we could potentially take advantage of these wonderful components and reshape and repurpose standard or new food products. And we've been working also in some cases in terms of downstream optimization. So assuming that you have a very nice product, how you can break down components, how you can actually extract what is valuable from that particular component in a much more, let's say, sustainable way. We're looking on a number of things around that. Supercritical sonication and enzymes are just an example of that. And then as soon as we consider that we have those ingredients, either microbiologically or from a plant, and how we formulate food. So what is the relationship between texture and microstructure, how these particular ingredients interact in that particular format and matrix, and how we could potentially incorporate them into food products and new innovative approaches. So in a nutshell, this is what I do. And hopefully we will get into new exciting things in the near future. And now I would like to present you a work that we did with one of our PhD students a couple of years ago, Zuhar Linda. So we were working on microbial astaxanthin production, and we were thinking which type of agricultural wastes are a bit more relevant for the UK and how we could potentially make that change and see how they can potentially work as substrates. So we were thinking about obviously astaxanthin, and we like yeasts in general. Astaxanthin is a pigment that it's been naturally produced by some microorganisms. It's found mainly in types of salmon and shrimps that are coming mainly from our diet. It's one of the things that we cannot really synthesize, so it has to be sourced from our diet. Has a high antioxidant potential. It is a precursor for vitamin A, and of course has a natural color. The main production of astaxanthin comes from a chemical synthesis, so there are a number of reactions involved in that particular area. And we were thinking how we can perhaps see whether we can produce them through a bit more of a biological route. So at the moment, there are two types of microorganisms, if you may, that they are much more, let's say, promising in terms of producing biologically astaxanthin. One is the algae Haematococcus pluvialis, and the other one that was the focus of our study was Xanthophyllomyces dendrorhous, or some of you may know it as Paphiarodoxime, which is the teleomer form of Xanthophyllomyces. And then in terms of substrate, apologies, before the substrate. So in terms of how this is produced biologically, I'm not going to bother you with a very complex metabolic pathway. Just going to do a couple of things for you, for your information. So the main pathways that are involved in the biological production of astaxanthin obviously are very complicated, but you have essentially through the isoprens biosynthetic pathway. So as soon as you have, let's say, a carbon source, a monomer, that's in our case glucose, and it goes down in the glycolysis pathway, it goes through the melavonate pathway and the isoprenoid biosynthesis, where you have specific precursors that are being formed at that particular point. And then they are lead into the final stages, which are the stages of carotenoids biosynthesis. Now, obviously, when we're talking about natural microorganisms and natural strains and wild strains, the expression of genes and enzymes involved can vary. And also the production of astaxanthin also varies as well. Generally, the biosynthetic route, the biological route, tends to have fewer milligrams of astaxanthin per gram of biomass. Still, when you compare it with wild strains, the chemical synthesis still is more, let's say, yields more astaxanthin as it is. But who knows? Maybe we will see some particular advances in that field, specifically when it comes into engineered strains. So the yeast, the Xanthophyllomyces dendrorhous generally produces astaxanthin, which is essentially a response to lipoprotein oxidation caused by environmental stress. So that could be oxygen, that could be other things. So naturally, we're trying to stress microorganisms, as it happens with most microorganisms in the future, in the nature, to make them produce the metabolite that we are much more interested in. Now, in terms of the substrate or the particular residue, we focused our attention on rapeseed. So rapeseed for the UK is quite a big crop in that sense. So it is one of the major oilseeds that's cultivated in the UK. The UK is the third largest producer after Germany and France. Quite a lot of arable land is being used for rapeseed cultivation annually, and its main product obviously is edible oil. So we have rapeseed oil on the shelves, but we also use it in other purposes for biodiesel and or as engine lubricants. So normally rapeseed oil is being produced by either cold press, so we press the seeds that are being produced, or we may have some solvent extraction in that particular case, depending on the use. However, the production of edible oil from rapeseed leaves behind a residue which is called rapeseed meal. Now, this particular meal, depending on the type of extraction that we have used, could contain quite a bit of oil. So roughly around 10 to 15% of oil is left behind in the meal in most cases, specifically when it comes to edible oil. And then the rest of the solids are mostly made up from protein and a complex combination of different types of carbohydrates. So you can have cellulose, you can have pectin, hemicellulose as well, structural proteins, and none of them interlink with lignin. So it is a very complex material, but because of that richness and complexity, could also provide, let's say, a very nice example of a byproduct that we could potentially convert it into a rich substrate for fermentation. So what we thought that we should do, so first we got the strain and we screened it on a variety of simple sugars just to understand how the yeast is being performing on a number of different single sugars. Then we proceeded with tackling the complex structure of rapeseed meal through enzymatic hydrolysis. We used a number of different commercial enzymes to see how we can break it apart and make it into production of a liquid that has assimilable free sugars and amino acids and proteins. So obviously we had to do that with a combination of different enzymes. So we used commercial enzymes that we're going to see in the next couple of slides, so that we can have a synergistic effect between different types of enzymes that are attacking different types of polymers, so we can break it down into smaller monomers, hopefully in the end. Then the other thing that we did is we went through the fermentation stage and we did some preliminary trials in flasks, and then we moved into a 2-liter bioreactor where we did a couple of different approaches in terms of how we can perhaps optimize a bit some parameters and see how we can rethink a bit the process that we apply in terms of optimization. I will tell you a bit more in the next slides. And then at the last stages of the work, we try to see how we can optimize or define a strategy in terms of extracting astaxanthin from the biomass. That was another point of focus of the study. Now, we did a bit of analysis of the rapeseed meal that we sourced locally from a local producer. And as you can see, we had probably around 17% of oil. So obviously, inevitably, we had to defat the material. But then, in terms of distribution, in terms of protein, we had a good amount of protein as well in the material, and then obviously a little bit of lignin as well, and probably 30% of sugars that after full hydrolysis into their respective monomers, we saw that they had quite a nice breadth and scope of simple sugars and monomers, which also essentially draw draw the need for us to be able to screen the yeast towards all those simple sugars to see whether all of them were assimilable by the microorganism. So you can see glucose coming in primarily from cellulose, but you can also see number of xyloses, arabinose, and galactose coming from the pectins, and then a bit more of galacturonic acid as well coming from the pectins, and then the hemicelluloses that they are part of the rapeseed meal. So we start off with the enzymatic hydrolysis. So first of all, we start off with viscozyme. So viscozyme is, again, a commercial enzyme that is very much used in lots of applications. So it's a multi-enzyme complex. So it has lots of activities, particularly in terms of breaking down carbohydrate polymers. But also it has, I think, a bit more of a slight activity in terms of protein breakdown. So we did a bit of an understanding and a bit of an estimation in terms of the enzyme concentration, and then how this was translated into concentration of sugars primarily, and then in terms of how this is impacting glucose yield. So we were focusing also on the glucose yield as well. Then we moved on into another enzyme, which is called pectinase. Obviously, then the focus here was polygalacturonase activity, tackling particularly pectins on that particular rapeseed meal. But this particular enzyme has also cellulase and beta-galactosidase activity. And then we stumbled across the fact that some of you, if you have worked with those enzymes, will see before. These commercial enzymes, they do have additives in place. So in the case of viscozyme, it has quite a bit of sucrose in there. In case of pectinase, this particular one from Novozymes, it had quite a bit of glycerol. So immediately, whatever we had in the enzymatic fractions were not really a good representation of the sugars of the rapeseed. Then we continued, obviously, and then we worked with accelerase. So this is predominantly an enzyme that attacks mainly cellulose. So it has an endo and an exocellulase activity as well. So although obviously the amount of sugars that we were able to extract were not that many, this was far more representative of some of the sugars from the rapeseed meal as opposed to others. And then we also work with the cellulase, which was an endoglucanase enzyme, which again, although it was not part of the specs of the manufacturer, it did contain quite a bit of glucose anyway in its particular formulation as as an enzyme. So using this particular information, we were very interested to work with accelerase, which we thought it was much more representative of some of the sugars that we get straight from the rapeseed meal, as opposed to having some additives from the enzyme as well. But we also throw into the equation a bit pectinase because of glycerol. We wanted to see having a bit more of a separate kind of carbon source there. Now, in terms of the growth of the particular enzyme, in particular yeast in simple sugars, so these were all trials that we did in batch plus cultures where we had different carbo sources. Apart from simple sugars, we also put cellobiose as a substrate, only thinking that we may have from the enzyme that we use some cellobiose being produced, so essentially two glucose monomers together linked. And as you can see, within all of these cultures, starting with 30 grams per liter, the yeast did pretty much okay, probably as expected. So literature suggests that wild strains of Xanthophyllomyces, dendrorhous, they mainly yield anything between 250 and 40 micrograms per gram of biomass. So we were probably spot on in terms of that. In terms of Umax and how well this particular strain can grow on these sugars, we had quite a good production in terms, quite good performance in terms of cellobiose, glucose, and fructose. The other thing that we saw was that we detected through the HPLC analysis of the broths ethanol, not a big amount of ethanol, anything between 1 to 2 grams per liter. But this suggested that we may have a possible thing that happens in some of the yeast, which is called the Crabtree effect. So some yeasts, after a certain level of carbon source, they switch their metabolism from respiration to fermentation. And then instead of channeling pyruvate in the TCA cycle, and of course the production of astaxanthin and carotenoids in the end, they channeled it into a different direction, which has primarily production of ethanol. So we were a bit vigilant about that to see how that will play out. So we did a bit of screening in terms of different concentrations of glucose just to see if this is a concentration driven process and where we have that particular onset of that metabolic shift. And we saw that around 30 to 40 grams per liter of glucose, we started off seeing this particular Crabtree effect. We kept that a bit as information for the future, as you will see. Now, we moved on into actually using the rapeseed meal hydrolysate as a substrate. So in these cases, we had no other supplements into the culture, so the liquid that we were producing after filtering the solids out from the enzymatic hydrolysis, we were using it as such as it was for fermentation. So as you can see, we tested all 4 different hydrolysates coming from the 4 different commercial enzymes, and we had varying particular performances. So in terms of viscozyme, we didn't see that huge amount of biomass being generated. There was quite a bit of ethanol being produced in this particular case, around 40%. So this ethanol here is denoted as maximum because we saw that after a certain point, and when the sugars were depleted from the medium, in some cases the yeast was able to consume some of the ethanol towards a bit more of further astaxanthin production. There was always perhaps half or 1 gram per liter left in the broth. Cellulase was by far, let's say, most in terms of ethanol production as well, but it did contain quite, let's say, quite a good production of biomass, probably in the expense of astaxanthin. Pectinase did not produce any ethanol, and that comes as to no surprise. Just remember that pectinase, as an enzyme formulation, contains glycerol, and this does not really initiate Crabtree effect. So actually, that particular yeast was able to tolerate very easily around 6 to 7 grams per liter of glycerol initially in the medium and produced quite decent amounts of astaxanthin. And then accelerase, on the other hand, had a bit of ethanol in there as well. However, there was not that much to begin with. And then in terms of astaxanthin yield, although the biomass was less than in other cases, it was much more enriched with astaxanthin. So we picked up from this particular exercise accelerase, and we worked a bit more with pectinase a bit for the future experiments. Now, the other thing that we did as well was to investigate a bit the aeration. So we know from the literature that one of the key things that drive towards higher astaxanthin production is aeration. So these experiments were performed with just glucose as it is, no hydrolysate or anything else, just to see, apart from supplying aeration, which was air at 1 liter per minute, how we can further help the production of astaxanthin by the speed of the impellers within the reactor. So we started off with 250 rotations per minute to begin with, and we also monitored the dissolved oxygen levels within the culture medium. So what we saw in that particular case was that we saw the rise of ethanol as it was, which was again slowly accumulated again and reconsumed again towards a bit more synthesis of astaxanthin. And then we saw that the dissolved oxygen was not really maintained after a certain point. Certainly anything that is higher than 20% of dissolved oxygen is considered more or less anaerobic fermentation. We had quite a bit of significant drop of dissolved oxygen during the fermentation period, so definitely 250 rotations per minute were not adequate. Then we moved on to 400 rotations per minute. They were apart from the, let's say, the peak of metabolic reaction and production of biomass and astaxanthin, we maintained DO levels above 20%. But again, that was not as good as we were hoping. So we increased a bit more the rotations per minute, and we reached up to 600, where we saw what were hoping for, that yes, you have a bit more of a reduction of dissolved oxygen when there is much more metabolic activity happening, but nevertheless, we maintained these levels above 40% throughout. So that was the choice for the next of the experiments. So maintaining in the bioreactor 600 rotations per minute, in addition to the aeration that we were introducing with air inside. Um, and then moving on into fermentations in the bioreactor, where we were able to control a bit better the fermentation. So we tested also cellulase pectinase and accelerators to see how these particular hydrolysates can meet all the requirements of the microorganisms. So the fermentation was quite quick. There was not, I mean, regardless of having some bit more glycerol or only sugars in there. And then in terms of performance, they were not bad at all, actually, very much supporting the growth and astaxanthin production. So this was essentially a very good indication that the hydrolysis of rapeseed meal actually can support on its own as a sole liquid in terms of nutrients, the growth of Xanthophyllomyces and the production of astaxanthin. Now, this is just an example of the kinetic. I think this is the one that we've used pectinase here. So you can see that the evolution here, quite interesting to see as well, was that the microorganism chose first to consume all the available simple sugars. So a bit of glucose there, a bit of arabinose there, and a bit of xylose there coming in from the hydrolysis of the materials. And then after these were depleted by the microorganism, apologies, after these were depleted, then the consumption of glycerol started, which obviously was channeled towards astaxanthin production. The other thing that possibly you could understand from here, but we also saw it in earlier kinetic studies of this particular microorganism, is that astaxanthin for this particular strain is a partially growth associated metabolite. So in the literature, you may have strains where astaxanthin is directly linked with growth. So when you have an exponential growth phase, you have also astaxanthin production. In our case, and for this particular strain, as soon as the halfway through the exponential phase, it's where we normally saw the onset of astaxanthin production. Now, all of these particular experiments, when it came into rapeseed meal hydrolysis, were done in the traditional, let's say, method of separate hydrolysis and fermentation. So in these cases, you have a vessel, whatever vessel that is, you put in your biomass, you add your enzymes, you allow for the hydrolysis to happen over a period of time. In our case, that was for probably around 24 hours. And then you separate the solids from the liquid. The liquid probably will undergo a bit of a filtration just to make sure that you ensure the microbial sterility. And then that is used in a bioreactor or a flask where you do the inoculation with your microorganism. So in this case, for instance, this was the example of the accelerase hydrolysate. So you can see that obviously we have all the sugars coming in from the hydrolysis, and then you see the production of astaxanthin. What we tried to do and see was that whether we can do everything in one vessel. So this is called essentially either a simultaneous hydrolysis and fermentation. We couldn't actually do a simultaneous one only because the enzyme that we were working on had an optimum of around 45 to 50 degrees, which that will obviously would be a killer for our microorganism. So we used one vessel. We introduced the biomass inside, we introduced the enzymes, we allowed for that particular hydrolysis to take place, and then as soon as we were ready to go, we reduced the temperature up to 30 degrees, which is the optimum for the microorganism, and then we inoculated. So we kind of mimicked that particular area, trying to do everything in one vessel. So what we saw in that case, obviously here we don't have biomass, we have colony forming units because it's very difficult to get biomass out of there. What we saw is that although we had an okay growth in terms of the yeast, we didn't really produce much of astaxanthin. Now, we came to the conclusion at that stage that because of the fact that you have too many solids within that liquid culture, the oxygen diffusion is not adequate. So even though we were trying to maintain that as best as possible with steering and the dissolved oxygen and the aeration that was going in, it was very possible that locally within the vessel there were areas that the solids were attaching to the cells of the yeast, and that creating some very, let's say, macroaerobic conditions which were not favorable in terms of astaxanthin production. The other thing that we did was try to do a fed batch approach. So a fed batch approach essentially is that when you are introducing your substrate in intermediately, you can also do it continuously if you're trying to maintain a constant concentration of carbon source. In our case, we did it in pulses, as we say. So in terms of glucose-based defined media, we did it as a trial to see how the microorganism will perform. And then also thinking in mind that we have that threshold of around 30 to 40 grams per liter of sugars that perhaps will trigger Crabtree effect. We like to keep that a bit less than 10 to 12%, 10 to 20 to 12 grams per liter when we were introducing that particular glucose in the substrate. So when we did that, we saw that actually we had quite a bit of a decent production of astaxanthin. Obviously, these were defined media, very much so. And then we did the same in the rapeseed hydrolysate media in accelerase, but using glucose as glycerol as a feed, only because glycerol, we saw that it's very well, let's say, received by the microorganism, and it doesn't really induce any Crabtree effects. To be honest with you, this was not very well optimized, and this was just a couple of trials that we did. I'm pretty sure that there is quite a bit of room for improvement here. We saw that there was a bit of a slower uptake of glycerol that we wanted to see. In terms of production of astaxanthin, these were probably, let's say, a bit less than in glucose-based media. There could be other strategies that we could potentially apply, so a much more continuous feed of lower amounts of glycerol could actually do the trick as opposed to just giving a pulse of 10-15 grams per liter. But there could be a possibility given the understanding that we have on how well this particular yeast grows on glycerol. There could be an opportunity here to take advantage of that as well and try to see whether we can leverage anything from that particular type of fermentation. Now, the other part that we were focusing on was, okay, we have a condition whereby we can grow the microorganism. We do have some good production of astaxanthin. How we get it out? So the focus was there obviously to use astaxanthin on its own as an ingredient, either for food or feed or pharmaceutical applications. So we went through the, let's say, the traditional way first, which is using glass beads as a way to break down mechanically the structure of the yeast cells. So for those of you that probably you have worked with yeast before or you've never worked before with yeasts, generally they are very much, let's say, difficult to break types of cells. They are probably the most stubborn ones in the microbiological world. So we used glass beads, and then we used a number of different type of solvents to see which one works a bit best. It came as no surprise that acetone and DMSO mixtures were the best. So astaxanthin on its core, it may have some aspects which are a bit more less soluble, a bit more soluble. More it is depending on which place it's adhered to. So it may need a combination of different types of solvents to have optimum extraction. But obviously we saw that with acetone, obviously this was the best. Acetone was also a reference method that we were using to measure the accumulation of astaxanthin in our cells. We did also a bit of a scanning electron microscopy just to see how the yeast cells look like before and after the extraction. So on the A1, you can see how the very nice yeast buds look under the microscope intact and just before being broken down. And this is what's left behind after glass beading and extraction with acetone. So you can see that you have quite a bit of a breakdown of cell wall structures. You can see some some particular debris of cells in that particular area. The other thing that we did was to see whether the accelerase enzyme that we used for the enzymatic hydrolysis of rapeseed milk can also be used to break down the cell wall structure. So accelerase, even though it has an activity, an endo and an exo activity of cellulitic activity, it also has some slight activity of beta-glucanase, which is essentially the enzyme that acts the beta-glucan structure of the yeast cells. So we want to see how accelerase would work. So we did a bit more of a response to phase approach where we wanted to optimize a bit the conditions, mainly in terms of temperatures and pH. And we worked with both dry and wet cells. So dry cells were dehydrated freeze-dry cells, whereas wet cells were just the pellet coming out shortly after the fermentation. So we saw that in the case of accelerase, we had a peak in terms of extraction, so around probably a pH of 4.5 to 5. That was probably the peak area, and temperatures nearly 36, 37 degrees. And the wet substrate worked better than the dry. Perhaps this doesn't come to no surprise because obviously there was a bit more water retained within the cells, and that possibly helped a bit more the enzyme work as well instead of actually rehydrating dried cells. We also work with another enzyme, which is an actual beta-glucanase. It's called, the commercial name is Glucanex. So an actual enzyme that has a beta-glucanase activity in high stakes. And then we also did a bit more of an optimization. So in terms of parameters, we were not very far away from where accelerase would work. So for glucanase, we had the pH of around 4.5, so slightly acidic pH, and a temperature of less than 35, which was probably a bit more desirable in terms of thinking about what is the optimum temperature for that. And then, as expected, wet conditions helped a bit more. And then here in these two images, you can see how the cells were looked before and then how they were looking after a Glucanex treated cell. So again, quite a bit of cell wall disruption in places, not very uniformly though. So obviously it depends on where the enzyme has more room to work. And then the other thing that we did was that we wanted to see how we could potentially extract astaxanthin with supercritical CO2. Well, at the time, we had another PhD student who was working on carotenoids extraction from agricultural waste, so peels and carrots and so on. So we want to see how the technology would translate within a biological material as is yeast cells. So we did a bit of an optimization when it came into supercritical. So these dots here are just the cells which were dried. So you can see the cells here, which were dried after production. And we had to use a bit more of a modifier because obviously we need to play a bit with the polarity to ensure a bit better extraction of astaxanthin. So in terms of polarity, we used ethanol as a modifier in the process. And then these conditions that you see were a bit optimized with another responsive phase methodology aspect for supercritical. But then we saw that the extraction, even in different, irrespectively of the stages of the extraction, so the steady state and then the falling rate and then the diffusivity rate, was not particularly improving. So after almost an hour and a half, we were probably a bit less than 20% in terms of extraction from the cells. So what we did is that we thought, how about first using the enzymes to break down a bit the cell wall structure and then subject them into supercritical CO2. Excuse me. So we work with accelerase and glucanex. So that 30% accelerase was the one that we were using in the previous responsive phase methodology. We went a bit higher as well to see if there is any improvement at all. Obviously, this is quite a lot of enzyme being involved in that particular case and not commercially applicable, but research-wise interested to know. And then that 5% glucanex was the percent of glucanex that we were working previously. So we saw that actually we had a bit of an improve, which suggests that once you are able to just penetrate a bit the cell wall structure, which which could be the primarily, let's say, barrier towards acceptability, then CO2 can solubilize and diffuse far better into the yeast matrix and can help a bit with the acceptability. So we reach probably around 50% of extraction from the cells, which was again on its own quite an improvement. Possibly there is quite a bit more to do with this particular aspect and see how we can leverage the technology and extract a bit more. Now, moving on from this particular work, what we are about to work on in the very near future is using this information, but now instead of focusing on extracting astaxanthin, using the whole biomass as is as a protein and color food ingredient. So we understand quite a bit more about the fermentation with this particular strain. So we would like to see in cases whereby we formulate or we produce meat alternatives, for instance, or meat analogs, how we could potentially use the color and the protein for these particular products. Color is a very difficult aspect in this particular field of innovation, it's very difficult to find a natural ingredient that sticks within the particular matrix and offers that color, which it is probably a bit red, that as we were hoping for, let's say, meat analogs. And then the other thing that we would like to see is that whether we could stabilize that particular color. So we've done a bit of work with anthocyanins, and we've seen that in the presence of some particular phenolics, we can have some intermolecular pigmentation, and we can stabilize that particular color. So we are looking forward in the next couple of months to see how we could potentially use that information to perhaps stabilize the color that we have from this Xanthophyllomyces biomass, so that we can formulate products and then extrude products that could look a bit closer to what a meat analog would resemble, hopefully. I think this is up for me. Obviously, acknowledgements. I couldn't. This is not. This could not be able and could not be realized without my good friend and colleague, Professor Dimitris Chalampopoulos, which we work very close together, and we share a group of very nice people. So this is the current group. We were off on a bowling night a few months ago. And on this picture, this is the former group. So this is Zuha, and the person who did the PhD. She is now an academic lecturer in Malaysia. And this is Michael. Again, he was working on supercritical back in the day. Now he's also a lecturer in Greenwich University. And these are Zuha's publications in case you're interested to look a bit more about this work. So thank you very much. And if you have any questions, I'm more than happy to try and answer them.
Thank you so much, Aphrodite. That was fantastic and very exciting to know that your current research is focused more on the whole biomass. And yeah, I think that seems quite promising as a research direction. The first question is there any reason why acetone can't be used during the supercritical CO2 process instead of ethanol as a co-solvent?
Yes, thank you for the question. So acetone, first of all, obviously it comes with a very low odor threshold. So it is not, let's say, a food grade solvent as we would hope. Ethanol is a bit more in comes to that. So I don't really remember the conditions that acetone enters into a supercritical state, if I'm honest with you. I need to double check you on that. But it's not a solvent that we prefer. It is, you will encounter it in, and let's say, baseline protocols when you don't want to have anything that interferes with your particular extraction or methodology, but it is not really something that you would use if, particularly if you want to develop a process for food grade applications. I hope I've answered the question.
That's very helpful. Thank you. The next question is asking whether the astaxanthin in Xanthophyllomyces is in free form or esterified. And
Yes, I think if I'm well, we didn't particularly look into that, but I think when it comes into this particular yeast, it normally comes in free form. It's not esterified. I think you will see it in the algae that it's sometimes esterified in lipids.
awesome. And then their follow-up question is, how is it possible to extract it at 360 bars in 90 minutes? What has been the CO2 to biomass ratio, if you're able to share.
The CO2 to biomass ratio. Let me go back into memory lane. So I think in the extract, so we have a laboratory extraction vessel system essentially. So I think the extraction vessel can take up to 60 or 70 grams of biomass. So that was the biomass in, and then the conditions we applied for CO2 were 50 grams per minute that we're pumping in a supercritical state. And then I think that the vessel that we have can go up to 400 bar. So we had a bit over optimized and we saw that around this particular pressure and temperature of around 70 also as it was, we were able to get as much as we could from the particular biomass.
Impressive memory. Thank you.
I did some
Um,
supercritical recently, so I remember the dimensions of the vessel.
yeah, I was like, wow, this is pretty profound. Um, Next question is, which stress conditions did you use to express AST?
So essentially, the stress conditions that happen is the nutrient depletion. So obviously, when you have some carbon being channeled towards biomass production, then when this been a bit depleted, this essentially triggers the production of astaxanthin. We didn't do anything in terms of adding more inducers at the time. There was a work that we did with the PhD student to see whether we can have some inducers at the. I think we used um, citric acid, if I'm not mistaken. We used a bit of glycerol and something else that now slips my mind. We didn't see any particular, let's say, stress-induced condition that really benefited astaxanthin at the time. These were all undefined media.
Very helpful. Thank you. If you have any more questions for Dr. Chatzifragkou, please add them in the Q&A section of your Zoom bar. I'll ask a high-level one just
Oh
because
yes, of course.
the week that we spent together in October and parts of your seminar just made me wonder whether you have thoughts on what you think the most neglected or underrepresented subsets of this research area are that you'd like to drive more attention towards.
Oh, now you caught me a bit off guard because I have to be honest with you. I haven't done my proper literature search for this particular topic, let's say very, very recently. So excuse me if there are any of the audiences that have worked recently on that particular area and I'm a bit behind. But I think that one of the key things that I think the area is about is probably focusing on engineering strains because even we understand a bit the biosynthesis of astaxanthin, the wild types are struggling to get high levels of astaxanthin. So I think there are very promising strains that are coming up in terms of engineering, either by direct mutation or another type of approach that they can really I don't know, perhaps produce us something 5 to 10 times more. So then we could possibly see perhaps a future whereby we have comparable production biologically as it is chemically now at this stage. So I think that it is probably that will unlock the commercial application of this particular product through biological production.
Awesome, thank you. So more emphasis on the upstream, either strain
Yes,
engineering side of things or the strain discovery that will just enable our basically production
absolutely,
host platforms. Yeah, that makes a lot of
absolutely.
sense. Thank you so much, Afroditi. This was a joy. I think we have another question that's just come through
Thank
before
you.
we
Keep them
start
coming.
moving towards wrap. Um, Perhaps I missed this, but do you have any rough cost or life cycle analysis for fermentation compared with extracting the astaxanthin directly from microalgae or crustaceans?
Very good question. I'm afraid I have none. And I have none. We don't do this well, we did back in the days we didn't do these studies only because when it came into production of whatever microbiological production of whatever component that you want, we were very much limited into a small scale, a 2 liter fermenter. So obviously this doesn't really translate into how the whole process, even from the upstream point of view, would look like on a larger scale. So we thought that we wouldn't be probably too accurate when it came to this. So I'm afraid we have no information about that. Hopefully in the future, because we were having some new developments in the department, we will be able to have some much more representative environment whereby we can do fermentations and get more accurate data so that we can do some life cycle analysis that is a bit more accurate and representative, more importantly.
Thank you. Yeah, it sounds like it's ripe for follow-on analyses. Another question here is whether you have any thoughts on how to determine biomass or growth kin in mycelium-based fermentation without destroying the entire batch.
So, well, yes. It depends on lots of parameters, so I need more information. So it depends on how you grow this. Is it a small scale? Is it a large scale? So normally when you do a small submerged fermentation, you and if it is, let's say, within a bioreactor, you inevitably you have to take a small aliquot and sample and then reflect that to the whole totality of the vessel. When it comes to solid state with mycelia, again, you need to take a small piece of it. So it's very difficult, and then you have to again to reflect that to the whole member of the biomass. So it is quite difficult to do that without some level of disruption because you need again to do some further cultivation on your samples to see how many spores you have or how much biomass you have. So it's a bit tricky. I don't know that.
Thank you so much, Aphrodite. This has been fantastic. Those are all the audience derived questions we have for today.
Excellent.
And as we drive towards wrap, I just want to give you an opportunity to say whatever it is that's on your mind to the global scientific community that we have with us. Is there anything that you'd like to share with this community as
Um,
closing thoughts?
Very nice. So thank you this. So thank you for that, Amy. I want to say that those of you that are interested in the alternative proteins community in general, I think this is now a time that this particular area of research has a momentum. Talk to your colleagues, talk to your peers, don't work in isolation. We have a lot to learn from different people who are taking just a slightly different approach, but everybody's working towards a different goal. I think it's important that we share our joint forces. Any type of experience is absolutely essential, and I think the scientists that you have in this particular community potentially will shape the future, our current future, in terms of how we see and do food systems. So yes, the future is in your hands, and it is transforming as we speak. So best of luck to you all.
Wow, I love that so much. Thank you for that inspiring call to action. You're so right that this is a multidisciplinary undertaking. It's an all hands on deck kind of moment for our food system. And thank you for galvanizing the scientific community with that reminder. Dr. Chatzifragkou, this has been a joy. You are so wonderful. Thank you for joining us and for your generosity in sharing your research with this community. To those in the audience, thank you for joining us today. Happy New Year again, and I hope to see you again next month for our next Science of All Protein seminar. Take care, everyone. Thank you.
Bye bye. Take care, everyone. Thank you.